I'm David Daniels. I teach in the university writing program. I'm not in charge tonight because I'm not a numbers guy. I'm a poet. I know that there are 2 beats in an I am, and 5 times 2, iambic pentameter. There are 10 beats in a line from Shakespeare. That's all I know. So I'm going to turn it over to some of my colleagues in a bit. However, the university writing program has a lot of important or big events coming up. In 2 days, we have another one of these faculty lecture series focused on qualitative research. We'll have a professor from anthropology and the director of the Civic Engagement Service Learning Program. Eric Fretz will be here, and Christina Krebs. And then we have a quantitative panel again on May 5th, and a qualitative panel, finally, on May 14th. And that will end our series of the faculty lectures. I also hold in my hand little flyers about upcoming poetry slams, open mic nights, including an open mic at Botanical Gardens that we've organized through the writing program. On Monday, May 12th, every DU student, faculty member, and staff member gets into the Botanical Gardens for free if you show your ID at the door. And there's going to be an open mic with students from DU as well as community members. So if you're interested in a flyer with a bunch of dates, come grab me after the lecture and I can hand you one of these, all right? At this time, though, I'll turn everything over to my colleague, Professor Campbell, in the writing program, and she will take over, all right? Thanks.
Welcome to the 3rd installment of our Researchers as Writers, Writers as Researchers series. Our basic goal here is to give some of the awesome faculty at DU a chance to share the content that they work with and their passion for their research while emphasizing the writing and research processes that they use in their work, perhaps to corroborate that we aren't just making this stuff up in the writing program. Tonight we're going to hear from several faculty members from fields that use quantitative research. And the faculty in the writing program are particularly excited about this because quantitative methods can seem pretty far afield from the text-based interpretive methods that most of us have any formal training in. While we do use qualitative and quantitative research in composition studies and education, most of us have to sort of figure it out ourselves. We have a few experts like Richard Colby, who will be hearing from tonight, who knows all about standard deviation, but most of us get a little bit nervous around too many numbers, so we're glad that we have our colleagues here tonight. Of course, quantitative research isn't just about numbers on the page, it's about a different way of understanding the world. And so that's one of the reasons why we investigate that as a separate epistemological tradition that helps to inform the way we understand and make meaning, and share that meaning both within the university and with the public at large. So a lot of times people will see these as very separate research traditions, and people will contrast quantitative to qualitative, but they often work hand in hand, and I expect we'll hear about that some tonight. For example, we might conduct an observation and notice a pattern, but in order to really see if that can be proven, we have to conduct a more detailed quantitative study. Or perhaps we notice a trend through quantitative survey, and we see, oh, well, people do this X number of times or with a certain amount of frequency, but through interviews we can gain a better understanding of their intention for that. And of course, those of us who work in texts all the time like to point out that most people do start from an understanding of the published scholarship to inform not only how they design their studies, but how they analyze the data. And so it's kind of hard to talk about any of these research traditions in isolation, but we're going to try. If you go to, say, Wikipedia and look for a concise definition of quantitative research, this is what you might find. Quantitative research is the systematic scientific investigation of quantitative properties and phenomena and their relationships. The objective of quantitative research is to develop and employ mathematical models, theories, and/or hypotheses pertaining to natural phenomena. The process of measurement is central to quantitative research because it provides the fundamental connection between empirical observation and mathematical expression of quantitative relationships. And so, if you remember important things about the scientific method, that comes into play here. But it's also about using numbers in a generative way and in a descriptive way to create new theories and then to apply those through further study. And so it becomes a cyclical process. Quantitative research is widely used in both the natural sciences, which are represented here tonight, and the social sciences. And we'll be hearing more tonight from the Department of Biological Science. First, we will hear from Robert Doris, who will be presenting some of his research about, now I'm going to do this again, the analysis of radiation of protenkephalin and protenorphin, that's it, protenorphin genes in fish, amphibians, and reptiles, as well as identification of novel opioid-like neuropeptides in the CNS of fish. He will make much more sense of that than I ever possibly could. That's why we have different disciplines in the university. Also, we'll be hearing from Dr. Doris first because he's lucky enough to have APS tickets tonight, I believe, and so he will bail on us shortly. We'll have a talk from him, and he'll entertain some of your questions before he has to depart and go watch some hockey. We will also be hearing tonight from Jennifer Hoffman, who's a professor in the Department of Physics and Astronomy. She uses combined techniques of observational spectropolarimetry. Yeah, that thing. And basically studying three-dimensional numerical radiative transfer modeling to probe the geometrical configuration of gas and dust surrounding stars and supernovae. So we'll hear more about that. We may be hearing from Darren Hicks, who's in the Department of Human Communication Studies, and we will certainly be hearing from Richard Colby, who is a lecturer in the University Writing Program, and he'll be telling us about some of the different quantitative studies that he uses in his writing class. So without further ado, I'm going to bring Professor Doris to the front, where he can explain, as I said, how he uses quantitative methods, and then we will have a chance for you to ask questions.
Okay, we just got to get the computer. Okay, well, I'm going to talk about the use of the scientific method. And the scientific method is incredibly straightforward. Essentially, what we do is make observations. And we make observations at several different levels. And it could be at the level of an ecosystem. It might be at the level of an organism. It could be at the level of a cell. It might be at the level of a gene. Now, what I've just walked through is a progression based on technology. If we're in the time of Aristotle, he could observe his environment. He didn't have much more that he could work with. Today, we can isolate genes, so we can look at them and analyze them. So technology influences at what level you can attack questions, but it still comes down to being curious. You make observations at different levels about things that you find interesting, and you identify phenomena that either are unexplained at this particular moment, they could be partially understood right now, or they are improperly described. Let me give you an example of the latter. Put your tongue to the roof of your mouth. Right now, you are right below your pituitary gland. It's about the size of the tip of your little finger. Now, Aristotle knew about the pituitary gland because they had plenty of cadavers from the numerous city-state wars that went on. So their anatomists had opened up bodies a lot. He knew what a pituitary was. It was obvious. It produced the mucus in your nose. Okay, well, that was all right. Now, that's 300 BC. Now, you go to Descartes during the Enlightenment. That's 1,300 years later. And he knows about the pituitary, and he goes, well, it's obvious. The pituitary is where the soul resides. Everybody knows that. Now, I'm an endocrinologist, and my discipline is about 150 years old, and during the last 150 years, we now know the pituitary gland influences growth, reproduction, stuff like that. So many things have been improperly described. And you observe, you make observations, and you say, you know, I think that they blew it. And so now you're going to correct things. And to do that, you first create a hypothesis. And the purpose of this hypothesis is it's a statement to help you resolve this phenomenon. And a hypothesis begins with, I predict. And then you make this statement, I predict the following. And from that statement, you will then design experiments to test your hypothesis. The better the experiments, the higher the probability that you will resolve your hypothesis. So, you evaluate those experiments, call that results, and there's a whole series of things that you might do. And then, you, based on the results, determine whether the hypothesis was correct, the hypothesis was incorrect, or the hypothesis was partially correct. It's usually the last one. That's usually what you get. Because the experiments that you're designing are taking advantage, once again, of the technology available to you, and they might not be able to actually resolve what you are after. So you have to take that into consideration. So once you've done the hypothesis testing, you've done the experiments, now you come up with a conclusion. That's really the big wrap-up. And that conclusion can lead you to the following. The first one is, you modify your original hypothesis. You accept that based on the experiments that you've done, you have to rethink what you're doing. Because it might have been the answer was your hypothesis was incorrect. So you can accept the data that's presented to you and say, you know, I need a new hypothesis. Or you can look at the data and say, I still like my hypothesis, to hell with the data. Now, that's the wrong thing to do, but actually that's what happens, I would say, about 80% of the time. That people will ignore the data because they like their hypothesis. So they'll start to muck around with it. That's bad. You can't do that. You have to step back now and say, I think I have to modify my original hypothesis. That's one thing. Now, it might just be that you have enough data from your experiments to say, you know, my hypothesis was correct. That's great. Well, that just means now there's going to be more of this phenomenon that you have to unravel. So now you will build a new generation of hypotheses to carry this story on. I have been doing this for over 30 years. Same problem, just keeps going step by step. So this actually works. Now, here's something that you have to be willing to accept. There is a point at which the current technology will not allow you to resolve the phenomenon any further. And then you say, I have reached my limit. If you go back 75 years ago, there were brilliant people who could not unravel basic phenomena in the life sciences because they actually didn't know what a gene was. So they had an idea that we had heritable traits, but they actually couldn't say precisely what that was. That's Watson and Crick in 1954 saying, actually it's these things called nucleotides, and they make up DNA, and on this DNA there are coding regions. Other people will say that. So there are limitations to what one can do. Now, the molecules that I work with are called opiates, and they're represented by things like morphine. That's derived from a poppy plant. And if you've ever been in pain, morphine is the drug of choice to alleviate that pain. Now, to be honest with you, it doesn't correct the problem. It simply makes you think that the pain isn't there. That's what any analgesic will do. But it gives your body the time to hopefully repair the damage. Now, isn't it amazing that humans respond to extract of poppy, and that produces analgesia? This led several people to propose in the 1960s that we, humans, and other organisms actually make our own endogenous, our own native opiate-like molecules. And this is what they look like. This is actually a polypeptide. It's called an enkephalin, and it has a shape similar to morphine. Well, these various opiate-like substances are encoded on different genes that are represented in these cartoons. You actually have 4 of them in your central nervous system, 4 genes that are being expressed. And the question that my lab has worked on is, well, wouldn't one do the job? Why do we have 4? How did we get 4? What is the origin of 4 genes that all appear to be doing something similar? So, given these observations, we have a hypothesis. And without getting into the details, the basic hypothesis is one of these genes came first. It was the ancestral gene. And then, over time, various duplications have happened that have given rise to the rest of the family. So this is a great hypothesis. But how do you prove it? Well, you have to choose some organisms to analyze. So I have a huge array of organisms to choose from. These are all our cousins. They belong to our phylogenetic group, the chordates. And they're everything from jawless fish all the way up to mammals over here. So we have a vast array of organisms to choose from to analyze, but I have one major problem. I want to know how genes have changed over time. The time frame is measured not in millions of years or tens of millions of years, but hundreds of millions of years. And I cannot go backwards in time. I cannot look at an organism that existed 400 million years ago and analyze its gene. I must look at contemporary organisms. So, by looking at contemporary organisms and comparing gene sequences, I will extrapolate back to what might have been. That is a limitation with any study that's based on an evolutionary hypothesis. Now, organisms that I focused on are right here. This is a better picture of them. These are called lungfish. This is the Australian lungfish. That's the African lungfish. These fish actually have a fossil record that does go back 400 million years. And they haven't changed much in the last 200 million years. Analyze their genes, and they may be a window to the past. That was our operating hypothesis. So, one of the goals of doing research, well, it's to publish paper. It's not only that we get a personal high from this, but it's through these papers that we are able to convince the government to give us money to run our lab. So there's a practical side to this. I have 103 publications. I'm editor-in-chief of a journal. Now, I've been doing this for over 30 years. So actually, that isn't very many when you think about that. But this is what we're after, to put together a paper that would be published. This happened to be in the journal Neuroendocrinology. This is a journal based out of Paris. And our papers follow the following formats, really straightforward. You have an abstract, and I'll tell you about each one of these. And an introduction, and then there'll be methods, and then there'll be results section, and discussion, and then acknowledgments, and then references, and then figures to illustrate what we've done. So those are the parts of our papers. And all of this will represent several $1,000s worth of research. And it all gets encapsulated into about 10 printed pages. But it was worth it because usually there's some interesting discovery that happens. So this is what an abstract should look like. The abstract is short. It's to the point. It may be the only part of this article that the rest of most of the people in the world are going to see if they even look at this. And so you have to tell them in a very concise way, this is what I wanted to do, this is what I did, this is what it means. The abstract is written last. It's when you've distilled everything. Now, the introduction. Notice it took up only one printed page. The introduction sets the stage. It says, here's the background information that you need to understand what I'm doing. The numbers represent references from the literature. Everybody builds on someone else. So, who preceded me that laid the groundwork that led to the hypothesis that I'll be testing? Now, I love it when an introduction ends with, and here's the hypothesis I'm going to test. And when I'm reviewing papers, that's one thing that I want to see in the introduction. I want to know what you're testing. And so that's a great way to end. That's the last paragraph. Then methods, I didn't bother to put those in, but methods are, here's how I'm going to do it. And when you read a methods section, you should be able to replicate everything that I've done. So that becomes critical. Now, in the things that we're doing, we clone genes. And so when you get a gene sequence, well, you have to convince yourself that you got it right. So you don't do it once, you don't do it twice, you do it about 5 times, and then you keep comparing the sequences to make sure that your machines aren't making errors, because they will. There is bound to be some level of error in the approaches that you are using. So you must have repetition so that you can say, I have a confidence level that I've got this right. One shot's not going to do it. And then you get to the results. This is actually probably the shortest part of the paper because it starts out, mine do, with a sentence, I'm doing this experiment because. And then here's what the data show. And there's a figure that will accompany that. and you try to be as concise as possible, and you do this, hopefully, in a logical way, so the reader is starting to anticipate, what are you going to do next? I bet I can guess. So that's kind of cool when that happens. And then you get data like this, which is just about meaningless to you, but all those letters represent amino acids, and this is an alignment where we're comparing several sequences, and from this, we can actually make sense of these genes and start to say who's related to whom. Now, you can do this with a computer, but actually your eyes are better. Humans are the best at pattern recognition. So actually, I don't use a computer when I do this. I do it all by hand. Because the computers initially were wrong when they were analyzing our gene family, and we got out a great set of papers saying, you know, the computers blew it. Because they don't think. They don't really think. They just assemble data. But we could say, you know, I'm going to be intuitive, and things started to fit together. So this is the payoff of all that work. Then in the discussion, here's where you summarize your results, and then you say, here's what this means relative to what everybody else is doing in the field. This is the wrap-up. Now, as an editor, I like it when a discussion ends with, and here's what I'm going to do next. I think that's great. A lot of journals don't like that. And so if I ever become an editor-in-chief, I'm going to do it this way. So my papers go to this chair. Anyway. But I like people to say, here's what we should be thinking about at the next level. My colleagues refer to this as speculation, and they're scientists, and they don't like that because they're precise, and we seek the truth. But I'll be honest with you, if you want to seek the truth, do philosophy. We're empiricists. We find out what we can know today with the technology available to us, and most of us have the humility to accept that we don't have the final answer. That it's going to be somebody after us, maybe a long time after us, that's actually going to come up with, and here's what this really means. I like to have this in my discussions. This is a summary figure that is actually testing our hypothesis, and in 3 papers, I changed this figure as new data said, you know, the old hypothesis needed to be adjusted. Now, that's what you have to do. Because if you keep saying, I will ignore the data because it doesn't fit what I originally said, you're going to end up printing stuff that is in error. So don't do that. And acknowledgments, this is to thank the people who paid for the project and who helped you. References, this is to keep people who are reviewing your papers happy because they like to see themselves cited. And it's the honest way to say, I'm not the only one doing this. There are other people that work on this. And then, of course, the illustrations are incredibly important. A picture does tell 1,000 words. And a properly illustrated figure will show your data in a way that will really bring it together for your readers, and that's what you want. So that's it. That's what I do. No questions. Okay. My wife is waiting for me at the Pepsi Center, so fire away. If you have any questions.
I have a question.
Fire away.
Bob, what's it like to work collaboratively? I mean, what's the research and writing like? I noticed like 6 people listed
Right.
on that. I just am not used to that.
Well, usually when we're working collaboratively, we're working with students, and several of the people on there were students. And then we will acknowledge people who have provided either critical reagents or have intellectually, well, they all have intellectually contributed to the project. So Hubert Vaudry provided the African lungfish tissue that we were working with. And we had talked about how to do this project in Tokyo 3 years before we did the project. So I felt he made a seminal contribution to getting us going. And then Jean Joss provided the Australian lungfish tissue. We have been working together since 1986, so we bounce ideas off. All of the authors review the paper and will go over, has the analysis been done carefully? Are the conclusions worthy? And then it's the students who do most of the grunt work of actually sequencing the genes. But working collaboratively is actually a lot of fun because you have people with different expertise who approach things from a slightly different perspective. And when you put very often you come up with conclusions that you probably wouldn't have gotten on your own. So that's why we go to meetings and we do a lot of stuff together. And thank God for email, because it's now opened up the whole world to us to bounce ideas off people. Remember to turn this computer off, because that's my count there. Okay, good. Take your time.
Yes, go ahead.
So much more clearly than I.
And I'm sure that will continue to be the case. And next, we're going to hear from Jennifer Hoffman. And once again, she's talking about things that I don't understand. So perhaps I should let her introduce her work again more eloquently than I can.
Yes?
No, it might just turn it off. There it is.
I think I'm ready to go if you're all ready to listen. My name is Jennifer Hoffman, and I'm from the Department of Physics and Astronomy, and I am an astronomer, so I'll be showing you some pretty astronomy pictures today. And I'm very pleased to follow Professor Doors. He gave a great introduction to sort of the basics of science, and I'm going to talk to you about, no, I'm sorry, it was a point. I'm going to talk to you about how I apply them in astronomy. And one really big difference between my work and Dr. Doors' work is he gets to do experiments on the objects that he studies. But I'm studying objects that are millions of light years away. So it's a very different kind of science. So let me first introduce the type of objects that I study. I study supernovae, and I study a particular kind of supernova called a core collapse supernova. On a very basic level, I mean, again, I like Dr. Doors' idea of kind of going back to the beginning of like, how do people first find out about these things? And if you can imagine, you're living in ancient times, and your culture is very focused on how things move across the sky, the repetition of natural cycles. You know when the moon's going to rise and set, you know when the constellations are going to change from season to season, and all these things are very important in your culture. And suddenly one night, everybody goes outside and looks up, and there's a star that they have never seen before, a new star that wasn't there the previous night. And all of a sudden you can imagine this is a big deal. This is an omen. This is something that is important in your life. And so it becomes a really sort of focal point for all this stuff. Maybe it has meaning. Maybe it means you've been doing the wrong things, you've angered the gods or whatever. But these phenomena that are called supernovae have been important to people since the time that we were able to observe them. And there are some that you can see with your naked eye. This is an example of a supernova that actually was visible with the naked eye from the southern hemisphere. It appeared in 1987, so a little over 10 years ago. And on the top there, on the left, you can see a picture that shows you before and after. Before, there's a normal star, and that's not a star you can see with your naked eye. That picture was taken with a very large ground-based telescope. And on the right side is the after. Suddenly, there's a much brighter star there. And our current understanding of these things is that they happen at the end of the life of a very massive star. The star is going through its life, it's burning, it's fusing hydrogen into helium in the core and deriving energy from that nuclear reaction. But at some point, the star's going to run out. There's only a finite amount of mass in the star. And so when it runs out, there's a gravitational collapse. The star does not have enough energy anymore to support itself against the inexorable force of gravity, so all of a sudden the thing starts to collapse, and then there's a rebound, because you can only squish stuff together so far, and at some point you can't squish it anymore, so there's a big rebound and a shock wave, and all of the material that was inside the star gets blasted out into space. That's a supernova. That's an amazing amount of energy and light suddenly appearing where there wasn't any before. And then if you look with a powerful telescope like the Hubble Space Telescope, later on, after the explosion, you see something like this on the right. That's called a remnant. That's what's left over. That's the gas that was inside the star that's getting distributed out into space. Go ahead and advance that for me. Give you another example. This is a supernova that exploded in 1054 and was recorded by Chinese astronomers and also, we think, by Native American population. This picture up in the upper right is from Chaco Canyon in New Mexico. And the position of that sort of starry-looking thing in the lower left with respect to the crescent moon, we think corresponds to the appearance of this supernova at this time in 1054. Now, if you look at the spot where the supernova exploded, nowadays, like these two pictures on the bottom with, again, Hubble Space Telescope and the Chandra X-ray Observatory, very powerful new technologies. Okay, as Dr. Doors was talking about, our technology has improved, so we can now see things that we couldn't see before. And here's what this supernova now looks like. It's now a bunch of gas that's being distributed through space, but still glowing, still very hot. The reason all these pretty colors are showing up in this picture here is that this is hot gas that's emitting light in various interesting wavelengths that tell you something about the state of matter in that gas. On the right, we have a picture in X-ray light. And this is the same scale, looking at the same object, but it looks very different. X-rays tell you something about a high energy physical scenario. So we're looking at very hot gas, very excited atoms, and the appearance of this thing should kind of look dynamic. It looks dynamic to me. It looks like things are swirling around and squirting out the sides. So what this tells you is even 1,000 years later, the supernova is long gone. You could not see this anymore. It was visible with the naked eye for a while, but now it's, there's nothing special with your naked eye. But still, at this point in the sky, there is very exciting dynamic stuff happening that we can learn about by conducting observations. So Dr. Gross talked a lot about observation, and observation is very key to astronomy. And so I want to get back to this question of how do I study these things? They are really far away. I can't do experiments on supernovae. I can't put them in a petri dish and see how they react to chemicals, and I can't poke them to see what happens, and I can't change things about them. All I can do as an astronomer is observe. Go first that for me. And the only thing I have to observe is light. Okay? This is a picture on the left of a big telescope. It's about 10 meters across. They're building it in the Canary Islands. We often speak of telescopes fondly as light buckets. And that has, light bucket really gives you a sense for how kind of passive this observational process is. All we can do as astronomers, experimentally, is sit around with our light bucket and wait for the light to fall on us. That's all we've got, because these things are millions of light years away, and we're relying on this tiny trickle of light that's falling, happens to hit the Earth out of all the entire universe, in directions they could go. So the reason you build giant telescopes is because the more light, the better, right? It's the same principle as doing your experiment over and over. The more information you can gather, the better your science will be. I want to say a couple things here about how we actually go about doing our science, because I think a lot of people don't realize this about astronomers. It used to be the case that astronomers would go to work every night, when the sun set, they'd go out to their little backyard telescope. This was before a lot of light pollution, of course. You'd kind of hike across the backyard into your little dome, and you'd sit there with your eye at the eyepiece and your sketch pad in front of you, and you would draw what you saw through your eyepiece. And this model continued for quite some time. In fact, if anyone has ever been to the Palomar Observatory near LA, that's a very large telescope, 100-inch telescope, where the astronomer used to sit inside the telescope tube at the prime focus where the light came to a point, and sit there with like a tablet in front of you, and the astronomer would ride in the telescope as the telescope would move across the sky. And my understanding is you couldn't get down, like it was a big production to get down, so you had to be sure you're not going to drink too much before getting into that thing, because you're going to be there the whole night. And you're literally drawing and recording what you see. So that's kind of an old model, but I get the feeling that a lot of people still think that's what astronomy is. Because I get people asking me, oh, well, you must stay up all night all the time. You're an astronomer, right? But that's not really how things work anymore. If I want to use a telescope like this, and this is kind of the cutting edge, if you want to use the best telescopes to do your science, you're not going to go out there every night. This is an international project. Lots of astronomers want to use it. So the process goes like this. You come up with a project idea, you write it down in the form of a proposal, and you send it to the committee that decides how to allocate the time on this telescope. So this is the first point at which writing becomes very important in my discipline. You have to make an argument for your experiment. You have to say, I want to know this, and here's my plan to do it using this telescope, and here's why it's important, and here's why only this telescope will do, and I couldn't just do it, at my local observatory that's just sitting in my backyard, or there's still a lot of local observatories, but these monsters are the ones that give you the best data. So you have to make an argument for how and why your project is important and why it should be funded. So then, if you get your time, usually you put in a request, I need a week, okay, of nights. And usually that gets cut down. So maybe you'll get 3 nights, right? And you might get 3 nights here and 3 nights there, and you might end up with a week per year of observing time. And during your week, or during your 3 days, you will often travel to the telescope and conduct your observations, but it's becoming more and more common that you don't even travel there, that you do it remotely by your computer. So somebody else is operating the telescope, and you're just kind of logging in to direct things. But even when you're at the telescope, you're not sitting there looking through the eyepiece. All of that is done, with computers now. We're using digital cameras, like the one in your cell phone, or like your digital camera, because those have a much better, in many cases, a much better response than your eye, and certainly a much better quantified response. So you take pictures that you can then read out digitally and store in your computer and analyze later. So the rest of your time, minus those 6 nights, right, is spent in front of your computer, working with your data, working with your images, and trying to transform that light that you've observed into useful information. Okay, go ahead. Move that one. So we have to be tricky with the light. We have only this one tool to use. So we have to try to extract as much information out of it as we can. Here's one thing you can do with it. You can take light and feed it through a prism, or what we call a spectrograph, which does the same thing. It spreads the light out into its colors. Okay, so there you get a nice little rainbow. Well, it turns out that when you look at a star or a supernova, you don't get a continuous rainbow, but you get one that has gaps in it. Either gaps or sometimes portions that are brighter than others. So you'll have, if you take this rainbow and plot it up as a plot that shows you brightness versus wavelength, which is the same thing as color, you're going to find that there is a sort of overall smooth shape, but sometimes there are dips and sometimes there are peaks. And those dips and peaks happen at specific wavelengths that correspond to atomic energy levels. So each atom inside the star, or whatever you're looking at, has a particular signature of these, what we call spectral lines, these dips and peaks, that will show up in your data. And you'll be able to say, aha, I see a hydrogen line here, a hydrogen line here, a hydrogen line here, so there must be hydrogen in this star. So that is a process of pattern recognition, again, as Professor Gore was talking about. You are looking at your data to try to identify the elements present. So that's one thing you can do with the light. I do something else on top of that. I study the polarization of the light. So let me tell you a little bit about what polarization is. You might know that you can think of light in terms of a wave. Okay, light is kind of a vibrating electromagnetic field, and it travels along and vibrates like this. Well, it turns out that if you have a light ray, you can think of it as a collection of waves, and they're all kind of oriented randomly. The vibrations are not all in the same direction. But sometimes something happens to the light that causes those waves to line up in the same direction, and that's called polarization. So on Earth, this often happens when light gets reflected off of something, like water or glass. And if you have polarized sunglasses, you can often see this, because the polarized sunglasses are letting through only light that's polarized in a certain direction, or not polarized in a certain direction. In space, there's not too many hard surfaces, so we get polarization by a process called scattering, and that's when light travels through a medium like gas or dust, and it requires polarization along the way as it gets bounced around. This actually happens on Earth too in the atmosphere, so actually the sky is polarized, and you can again see that for yourself. You take your polarized sunglasses outside, and what you're going to do is look at the sky and kind of rotate your sunglasses around and see that the sky gets brighter or darker depending on the orientation of your sunglasses. And the key point here for what I do is that the amount of polarization produced depends on the scattering angle, and that is just the angle between the original path of the light and the final path of the light. So it comes in, it hits something, it scatters, it goes off in a different direction. And the amount of polarization produced depends on that direction, that change in direction, that angle, scattering angle. So what that means is, if we measure the amount of polarization and the orientation of it, that gives us a clue to the geometry of the scattering region. So this is really powerful. It gives us an idea about what the shapes of things are in astronomical objects, again, millions of light years away, where we can't hope to see them with a telescope. With a telescope, all these things look like are points of light. Okay, you're never going to see it with your eye. But this kind of analysis lets you get a sense for what the shape is of that object that you can't see. One more just quick illustration of this. If you look at a round object and the light comes from the middle and scatters toward travels toward the outside and then scatters toward you, you're going to see unpolarized light because all those polarization vectors are going to cancel out. Okay? You're going to get no more polarization in one direction than another. But if you have a flattened scattering region, you have what we call incomplete cancellation. That just means that there's more polarization in one direction than another. And so we can actually extrapolate what we observe here. If we see light polarized in a particular direction, we can extrapolate from that and say, aha, this light must have scattered in a flattened distribution of gas or dust, and we can actually figure out what angle of orientation that scattering region has. So to get back to my objects that I study, we actually observe that supernova light is polarized, which tells us that supernova explosions are not round. So we kind of think of stars as big round balls of gas, right, like the sun. it turns out that that's not really always the case. Sometimes stars are surrounded by gas and dust that they have either accreting or maybe throwing out. And similarly, a star when it explodes doesn't just become a big round fireball. It actually has a shape to it that's more complicated than a sphere. So you might have guessed this already because I showed you some of these pictures, and you can see in these remnants of supernovae that they're not round. They're very highly not round. And so this might suggest to you that the explosion itself, which we couldn't really observe because it was too small, might have some shape to it as well. Now, these are examples of, these are not supernovae. These are stars, massive stars, that have, well, okay, so the one on the left is not a massive star, but these are other kinds of stars that are surrounded by circumstellar material that show non-round configurations. So we have several lines of evidence that suggest to us that there are interesting geometrical effects in supernovae that we can hope to observe. And part of my research is looking at how an object like this one on the bottom, which is a star undergoing an outburst, but not a supernova yet. This thing will be a supernova eventually. And I'd like to make a connection between a star like this and then what we see from an actual supernova explosion. So there's one other step to my research. Go ahead. I told you about the observational step where we're collecting the light. And then the second tool that I use to study objects that I can't see is I make computer simulations of them. And this is the experimental step. This is the part where I actually do get to kind of poke things and see how they respond. But what I'm doing is poking the model that's inside my computer. And I'm using that as a substitute for the object that's out there millions of light years away. And this picture on the bottom is just some simulations, an example of some of the simulations that I've made in the past. So this, I will try to keep this not very complicated. But what my computer model does is it pretends that treats light as a bunch of little particles. We've kind of gone from the wave picture to the particle picture now. But what I do with the computer is I say, all right, suppose we have a non-round shape, and maybe it's an ellipsoid, or maybe it's a disk, or maybe it's a jet or something. I'm going to put in a geometrical structure into the computer to just describe it mathematically. And I'm going to say, okay, here's the scattering region, and this is what I think is going to polarize the light. And then I tell the computer I'm going to put a source in the middle of it, and that's where the light will come from. And then one by one, the computer will emit virtual photons, light particles, and have them travel through the scattering region and come out the other side. And then what I do is take that. I've got a 3D picture in my computer, and I pretend to observe it with my telescope. And I say, okay, if this is an accurate representation of reality, of the supernova, what would I observe? And then does that match what I actually do observe with my telescope? The point of these models is to kind of make a connection between the physics that we know from the laboratory and from the experiments, the things that you're learning about in your physics classes, basic knowledge about the nature of light and about the nature of matter, and translating those into predictions that have implications for the large scale. So this is where the interface happens between theory and observation. Okay? This is how we're translating what we know about the universe into how we can say something about objects that we don't know about. So there's kind of an assumption involved here that our laws of physics that we think we understand still hold true out there 100 million light years away. So, I mean, that is maybe a basic assumption that we all share, but kind of, it's an assumption we have to make, otherwise science can't proceed. And it actually turns out that if that assumption is shown not to be true, it's usually a problem with our scientific theories rather than a discrepancy in the way that the laws of physics actually behave. One interesting point is that this kind of modeling requires a lot of computing power. So I use supercomputers, and sometimes takes them a whole day of running on a supercomputer, maybe 100, maybe 500 processors to run a very complicated model. And so this kind of stuff, this is another example of the kind of technology we didn't have even 10 years ago. So the kinds of models I'm able to make are very much a function of the state of the current technology. And in fact, some people when I started my PhD thesis, some people thought this kind of modeling it's not that interesting, it's not that useful. It's too much computer time for too little output. And it's actually proving not to be the case, because computing power has increased so much recently that it's pretty cheap. You know, I can run simulations essentially on a desktop now that can give really good results. Okay, yeah, go ahead and advance that, and I'll get done. This is again my technical slide, and I want to just show you this kind of comparison of observations with simulations that I'm doing. So on the upper right, in purple, is an actual observation. That's data from a supernova, and this is a region of the spectrum. Okay, so remember we had spectra that I could tell you the amount of light at a specific wavelength, and here's a region of the polarization spectrum. This is the polarized light, and it's showing you a little peak at a specific point in the spectrum. And I'm interested in that shape. I want to say, how can we explain the existence of this shape? This is a hydrogen line, and it's telling us something about how the hydrogen in that supernova is behaving, its velocity, its temperature, its density. Okay, so these are things I'm trying to reproduce with my model. Now on the bottom are two different kinds of outputs from my models. And the shape, the little ellipsoid and the little ring, represent two different models where I said to the computer, okay, let's see what happens if the light, if this polarization is produced by scattering in a ring or produced by scattering in an ellipsoid. And then the different colors are different viewing angles, so you can take a computer model and look at it from here, and see how the data change. And the little black lines are error bars. So we heard about uncertainties in your data, and these are things we have to include because the computer model has some limitations of its own. But basically what I'm doing with this is trying to figure out what combinations of parameters I can put into the computer to roughly match what's coming out of my observations. And you'll notice they don't match super well. The ones on the right match better than the ones on the left, but I'm still not actually creating a really great match. But going through this process allows me to narrow down the range of parameters that I can study. I can actually say, well, maybe I haven't exactly matched it, but I can at least say the density is between these two values, or the temperature is between these two values. And I can get an idea of how different physical parameters, different physical characteristics of the system produce specific kinds of observational output. And it's all essentially in the service of understanding these objects better. So my work basically walks the line between observation and theory by using this model to kind of negotiate between them. Okay, one more, I think. Oh, yes, and so, two more. So then after doing this kind of analysis, one comes up with a picture, an explanatory model, where you can say, well, okay, based on my observations and my modeling, I think that maybe this supernova has this kind of structure, for example, where you have the explosion, the part that's exploding in the middle is kind of elongated, and then it's surrounded by a flattened configuration of gas that maybe was there before, and then the whole thing is surrounded by some further gas, and we're observing it from this particular angle. So this is the kind of thing I might put in a paper as the conclusions that are suggested by the model. So one more thing I wanted to tell you about is what kinds of information I have to put into a paper to make it persuasive. So when I get to the point of writing my results or presenting them in a talk, I have to include certain kinds of information to make it persuasive, right? So that other scientists will say, oh yeah, that was a good job. This person has done a good job, and I'm convinced. Or they might say, if I haven't done a good job, they might say, well, I don't agree with the way that you conducted this analysis, and therefore I have the following problem with this. And then they might go and repeat it or do it better and publish their own paper. So first step is the observations, right? The light bucket thing. It's kind of ironic that even though we have all this technology, these very advanced, super large telescopes, and these fast supercomputers, we're still very much at the mercy of low-tech things like the weather. I mean, literally, if you're at a ground-based telescope and the sky is cloudy, you're out of luck. I mean, unless you're doing radio astronomy, which doesn't care about clouds. But I know people whose entire projects have been derailed by a run of cloudy nights. You just have to deal with it. There's nothing you can do about that. And of course, there's a middle middle ground between completely cloudy and completely clear. So in the middle there, you have to talk in your paper about what was the weather like. Was there fog? Was there scattered cloud? What in the weather might have affected your data? What instrument were you using? What telescope were you using? Which night of the year was it? And all of these things might affect your data and be important in somebody else assessing their value. You have to talk about what you did to convert the light you observed into sort of physical data, because that's another step at which you might be introducing uncertainty or assumptions that other people might not agree with. Dr. Doors talked about referring to other people's work, so I won't do any more of that, except to say you always have to do it, because it makes it, it convinces other people you're not just some crazy kook who's coming up with these ideas off the top of her head, right? Other people have had these ideas, other people have shown that this works, here's how my project fits in with the currently accepted theory. I have to talk about my computer program very specifically. I have to say, here are the assumptions I made, here's what the computer is actually doing, here's how we take the output and analyze it so that, in fact, many people make their computer codes public so somebody else can download it and use it for their own data. And I have to compare with accepted situations. I have to say, well, okay, so my code does reproduce, for example, currently accepted results that produced by trusted computer codes in the past. So again, I have to show where this is coming from, how it relates to stuff that's already happened. Error analysis, we talked about a little bit. You always have to be scrupulously honest about the sources of uncertainty in your data. Because otherwise, your data essentially have no value if they don't have errors on them. You always have to include some assessment of how, well, I guess Dr. Doors said it very well. It's a statement of confidence in your data, okay? It's an assessment of how good the results are, right? I'm 90% confident that this measurement I made had this value, but it might be a little bit off one way or the other. So you always have to have that qualification. And people judge your work not necessarily on the errors being small, but on the errors being correctly assessed. And then finally, when you get to the point of creating this sort of overall big picture, here's what this all means, you have to be very clear about the assumptions that you've made in drawing those conclusions, because again, this might not be shared by all your colleagues. So one more, I'll just leave you with another pretty picture, and make the point that supernovae do occur in many different kinds of galaxies, nearby and far away, and the most astounding thing about this whole process, the whole idea of studying astronomy, to me, is that we can actually do it. It's pretty amazing if you think about the big picture. We're stuck on this little rock, orbiting this little old sun out in the backwaters of the Milky Way, and we can, just by sitting here and just by looking, come to these conclusions about the universe as a whole. It's astounding, and it's really exciting for me to be part of.
I get one again.
All right, you go
Because I love this stuff. I don't understand stars at all. But they're pretty. Something you said at the end, I want you to repeat for my students. You said people judge you not on your errors being small, but on the accuracy of how you assess your errors. And I tried to convince my students today that if their experiment fails, it's still possible to write in a paper. So that there's not a relationship between the experiment falling apart and how they write.
This is very true. There's a concept in science called the null result. And unfortunately, we get so focused on the flashy result where you say, yes, hypothesis confirmed, that we forget a null result is equally important. And the null result is when you don't confirm anything. Maybe you don't disprove anything, but you don't prove it either. It's just kind of, okay, we did the experiment and we don't know. We still don't know. The value of those kinds of things is that you can often, just by virtue of doing the experiment, push back the limits of what we know a little bit. Okay, so maybe you haven't completely answered the question, but you've staked out some more territory that we didn't know before. You've done an experiment that hasn't been done in the past, so you have kind of explored one more little corner of what we call parameter space, and that's kind of the whole range of possibilities that are out there. So even if you don't get an answer, you have still produced, some value towards science. And, I think in science we do get focused on the big results, the big names, the flashy stuff, whereas really the whole field progresses very slowly, and it's a matter of many, many people doing many, many experiments and laboring in obscurity for years. But it's the way the field advances in very small steps. I mean, so even if your experiment doesn't do what you wanted it to do, you've still made a contribution. As far as the errors go, I would be suspicious of small error bars, right? After you've done this for a while, you get a sense for what the errors should be, what the uncertainty should be for a given experiment. And if somebody publishes a paper where the errors are very small, that's suspicious. Like, well, I think maybe you haven't accounted for everything there, because there certainly are more ways in which you might have uncertainty than you are telling me about. And so, as a scientist, I would find that suspicious. Okay, I would suspect that maybe you are considering something. So. Really, you need to be very careful about all these things. And a lot of the sort of bickering that goes back and forth among scientists is about, have you correctly accounted for this? Have you correctly accounted for that? So yeah, the devil's really in the details, but I would stress to your students that it's ultimately not about whether the experiment succeeds or fails. It's about what can you learn about it anyway.
Good, very well.
Thank you very much. Follow-up question? Thank you so much for your time.
Are you going to need this, or are you just going to talk?
I'm just going to talk.
Okay.
And all my students who have me past or present are going to be thankful because they seem not PowerPoints. I'm going to read part of this, but I have a preamble of sorts before I start. Some might find it interesting, some might, but some might not. Quantitative research in writing studies is burdened with much critique, some of which is quite valid, but most of which stems from a fundamental flaw in educational research. That sense may be a bit difficult to understand, even if you understand all the words that I said. One problem is that we don't normally associate the study of writing or writing classes with quantitative research. I could ask you all, have you ever been in a writing lab where you studied the number of particular clauses or sentences in your writing as Bunsen burners floated next to you? Or have you ever had a writing textbook that did a statistical analysis of what good writing was and next to microscope images and scatter graphs? Probably not. Another problem with my sentence is that idea of writing studies, a term some of you might not really be familiar with. For the record, writing studies is the study of the processes of writing, the effects of that writing cognitively and effectively, and understanding of its effects on readers. Yeah, I know, more jargon. Basically, writing studies looks at writing and writers. Returning to my opening sentence, I have one last issue to deal with, the fundamental flaw of educational research. One of the earlier educational researchers, Fred Kellinger, first introduced the flaw in the 1960s, saying that educational research is often short-sighted because it's looking for that immediate result. I want the immediate result. I want that practical thing I can use in the classroom. Whether we want to blame that on the American philosopher or much earlier educational researcher, John Dewey, for this flaw is a matter of debate. But for now, I'll put it simply that if a study of writing and writers doesn't deal with the immediate and pragmatic results in the classroom, it is often and arguably unfairly critiqued by those at the front of that classroom. So far, I've talked for a few minutes, and I've only mentioned the terms quantitative research a few times. In my defense, though, because all the reasons that I've just described, I cannot just talk about the rich field of quantitative research in writing studies without acknowledging that many, inside and outside the field of writing, distrust quantitative research of a topic that many consider to be an artistic act. Now, for the interesting part. When I was in college, many years ago, as a freshman, I stumbled across what I thought to be a really cool article. It was an article that counted the uses of second person in Shakespeare's plays. And second person is the use of you, but in Shakespeare's time it was thou and thee, so it counted all these uses. Since none of my professors had ever mentioned this, I thought it was a unique field. This seemed like real evidence to me. I thought, rather than try, work really hard and look for the meaning of white in Melville's Moby Dick, I thought to myself, I'd rather be able to just say, the word white is mentioned 275 times. Sure, it makes absolutely no sense, but to an English major vexed by the various interpretations that seem to come so easily to my professors, this seemed as relevant an answer as any. It turns out people do these types of studies, and this unique counting of second person in Shakespeare or sentence length in Faulkner and superlatives in Dickens is a relatively common thing. It's a type of quantitative research, it's that type of counting of a phenomenon. Now I could narrate how I later encountered linguistics and discourse research from the counters, the quantifiers, and how these were, again, unique fields rather than the mainstream. But I want to bring this around to writing studies, my work, and why I mention any of this at all. Writing studies tends to focus on academic and professional writing. By professional writing, I'm not talking about writers who write for a living, the Stephen Kings, the Dan Browns, but I'm actually talking about the writing in professional settings. Eventually, you're going to have a job, you're going to be expected to write in that job, and that's really what I'm looking at. But also, of course, academic writing too, both students and the writing of your professors. What process and rhetorical approaches a professional or academic writer considers, how he or she crafts a text, and what that text ultimately communicates is what interests me in English studies. And what interests me the most, however, is how we can teach those things to students. To that end, writing studies became a way to inform what I do in the classroom, and it's of course the reason I got into my field, my degree, rhetoric and composition, as opposed to astronomy or biology. Besides that perception of writing as an art, another reason that quantitative research in writing studies, just like in other strains of English studies, is not in the mainstream is because writing studies tends to be the domain of those who teach writing. Now, as I mentioned earlier, that educational research flaw tends to create problems since counting the number of correlating adverbs in a professional memorandum is a bit removed from the actual teaching of that memorandum in the classroom. After all, just because a professional memo uses 3 howevers, 2 consequentlies, and a therefore, doesn't equate with me teaching students that a memorandum generally should have 3 consequentlies, 2 howevers, and a therefore. In fact, one trajectory of quantitative research that has been abandoned in writing studies was the T unit. The T unit, or terminable unit, consisted of counting the length of main clause and its subordinating clauses. Sure, I might say a T unit of 14 is better than, say, a T unit of 4, but given real writing in a real rhetorical situation, that T unit tells me about as much about your writing as, say, how many consequentlies you use. So let me talk about one real specific thing, a more tangible way you can look at quantitative research and how I use quantitative research. We are currently conducting a longitudinal study of writing here at the University of Denver. And we're using a variety of different instruments in which to look at this. Although purely a descriptive study, and that's a very important distinction to me, we are collecting data that can be read interpretively, qualitatively, and quantitatively. There's a multiple choice questionnaire, an item that is usually used for quantifying the experiences, attitudes, and beliefs of its participants. We have interviews to follow up to collect qualitative data, and we are collecting student writing to look at interpretively. What I want to emphasize in descriptive writing studies is that each method can be read in multiple ways. For example, on the questionnaire, we ask both multiple choice and open-ended questions. The open-ended question could be assembled just as a large bin of qualitative data to help us better understand student writers, or we could code the data. In research, to code something is to apply a label to an item that could also be applied to other similar items. For example, we might ask students about their writing processes and then might choose to discuss writing, on the computer. We then might code that as writing medium. That's what students chose to talk about. Which would be different than, say, if a student discussed how or she assembled each part of the writing that they were doing. In other words, we are categorizing different responses so as to more easily count those responses. Such categorizations help us to also tailor the multiple choice questions in the future, since we have piloted the questions and received a sample of responses. The same could be said of student writing. Looking at the writing, we might all use interpretive methods that analyze the overall approach to audience using a rhetorical framework, such as logos, ethos, and pathos. However, we could also count the number of comma mistakes, count the instances of second person, or count how many times you mentioned white. In other words, although the writing lends itself to interpretive research, given that it is a full text written with a purpose, we can also approach it from a purely quantitative means to say that, in fact, students don't make all that many more grammatical mistakes than professional writers. Sometimes also, we have to quantify large sets of raw data just to make sense of it. We have conducted 30-minute to hour-long interviews with almost 100 study participants. As you might imagine, it's a lot of interviews and a lot of text. So reading through the interviews, looking for words or ideas to count and classify helps us to be able to better sort through the data and look for patterns. Now, in an experimental or quasi-experimental study, this sort of after-the-fact quantification of data would keep professors Hoffman and Doris up at night. However, for us in our descriptive writing, descriptive studies, this data can give us a simple measure to help us talk about writing as well as help us affirm or revise our program goals. And I'm going to stop right there. I can tell you about World of Warcraft. Shall I talk about it? Okay. So currently, I'm teaching a class in World of Warcraft, as is my wife. And one of the things that I want to talk about really fast is my approach to teaching and how these quantitative studies can also inform my teaching. One of the things that the lens from which I come at teaching from is a theory by Annette Karmelow-Smith. And she talks about these three phases that learners go through, learners. The first is behavioral mastery. Anytime we run into something new, we want to learn it first. We want to, in some ways, emulate how other people are doing it. And so we might imagine an assignment that a copy will follow the formula in it. But one of the things as a teacher is that I recognize that y'all can write. I'm not teaching you how to write. Y'all you made it into the university because you somehow could write and could write for a board. What I'm trying to get you to recognize as an instructor, as many of my colleagues are doing, is this idea of moving from representational redescription. It's a tongue twister if you ever heard one. In other words, being able to think critically about the writing that you do, and moving into what's called conscious access. Being able to say, oh, I am using logos here, or I am using the IMRAD method here because the rhetorical situation calls for it. Not because I'm making you do it, but because you recognize as a learner and a writer that the situation calls for this particular type of writing or this particular type of style. So one of the things that my wife and I, when we designed the World of Warcraft class, the ideal of it is not that we're going to teach people how to play World of Warcraft. They know how to play World of Warcraft. Nor are we going to teach them how to be writers. They know how to write. They know how to write. They've made it into the university. They must know how to write. The idea there is to get them to that third stage, that idea of getting them to be consciously think about the rhetorical choices that they make. So they get to pick their own research methodologies. They get to pick their own writing strategies. And the reason for that is because we theorize that we'll better get them to critically think about their writing not only in this class, but in their other classes as well. They're not just emulating behavioral mastery, but they're thinking about rhetorical situations more broadly. So that's my World of Warcraft part for you.
And do we have any questions?
And I'm sorry there's no PowerPoint. I should have just played World of Warcraft. Oh, yes, yeah, sorry.
So when you present your results like at conferences, do you still get people who kind of denigrate it, saying, well, what can you really learn from this kind of quantitative analysis?
I think given the variety of rhetoric and composition, people tend to just not go to these. You know, it's one of those things where if it's that session, they're like, oh, they're doing that. I'm not going to go to that because it's on numbers. I'm going to go to the, you know. Yeah, sorry. How does qualitative research help you think critically? Quantitative. Let's first of all, we'll define critical thinking, okay? Because you hear the term a lot, right? Okay. Yeah. Critical thinking, and this might sound just like circular reason, but this is the definition of it. Critical thinking is having a set criteria by which you're going to judge something. My technical term that I use quite often. I'm going to judge something. For example, let's judge a movie. I'm going to have a list of criteria by which I judge that movie. Whether that criteria is good acting, or nice camera work, or great action scenes, interesting, that might be my criteria, okay? So what all critical thinking is applying a set of criteria to some other thing that I'm looking at. Thus, if we're going to look at quantitative research, I'm going to have a set of numbers by which I'm going to judge, or I'm going to have a set methodology of research by which I'm going to judge this thing. For example, if we look at astrophysics, right? Professor Hoffman has a set of criteria by which he is going to judge whether her hypothesis was answered or not, okay? And that, in some ways, she's engaging in a type of critical thinking. And for me, as a teacher of writing, what I am trying to do is get students to generate their own type of criteria by which to judge a research situation. And by presenting quantitative research methods or qualitative research methods or interpretive researches, it gives students a sense of, oh, that's the framework by which I should approach this research. Or, in other words, I'm going to generate this type of research method. Does that answer your question? Okay. Nothing else? Everybody's ready to go home? Yeah. Thank you.